Hi everyone. This is going to be a little different than our regularly scheduled programming here, but I had to take a bit of a hiatus. As it turns out, finishing a PhD is very time consuming, and I had to focus on finishing up my revisions, writing my dissertation, and ultimately finishing my PhD. So I had to take a couple of months off here, but I'm excited to get back into recording a lot of these tutorial videos. But first, I really wanted to show you all what I've been working on since I took a little break here. And so here, I'm about to show you my public PhD dissertation defense. And this is going to cover two main projects I had done during my PhD, including a work focused on therapeutic discovery in Wilms tumor, as well as a second project looking at variant interpretation in a gene called SMARCB1. So, yeah, like, super excited to kind of pick this back up now that I'm done with my PhD finally. Yay! But yeah, this is kind of a more biological take, and you can really see the big picture here on two separate projects and how genomics can be used to actually answer biological questions. So, yeah, without further ado, enjoy the video.
All right, well, this is very crazy and surreal. So, good afternoon, everyone. I want to thank you all for being here this afternoon. You'll probably know, but my name is Garrett Cooper, and I am a graduate student in the Genetics and Molecular Biology program. As Andy just said, I've been in the Hong Laboratory for almost 5 years now. And during that time, I've focused on advancing diagnostics and therapeutic approaches in childhood cancer. And so I'm really excited to share the results of my graduate today. And so during my talk today, I'm going to be covering two challenges in pediatric oncology, but I would say also in the broader oncology field. First, we as a field have struggled to understand how specific genetic variants function within cancer cells. And second, we have struggled to understand or identify targeted therapeutics for cancers that desperately need new therapeutic strategies. And so, to contextualize these two problems in the broader field of the cancer research, I'm going to bring you back to the year 2000. So, funny enough, I was actually only two at the time that this magazine came out. But this Time magazine cover really captured the hope and the promise behind the Human Genome Project at that time. The promise was that now that we're armed with the genetic code, scientists would be able to revolutionize how we diagnose and treat all sorts of human diseases, and I would also say particularly cancer. And so, in many ways, the promises of this have actually been fulfilled. So the early 2000s and 2010s saw a lot of successes. There was widespread sequencing of patient tumors, and this ultimately led to the identification of specific kinases, particularly EGFR, ALK, and BRAF, as key drivers in specific cancer subtypes. And due to this discovery, scientists were able to develop small molecules to directly inhibit these mutations. This ultimately led to increased survival for patients with these particular mutations. And so, with these early successes, this led to this new precision oncology paradigm emerging in the early 2000s. The idea is that we would sequence a bunch of tumors, we would identify the driver mutations, and then we would target these mutations with drugs. It seemed like a simple solution to cure cancer, right? Well, as a lot of you know, in the science world, biology tends to be a lot more complicated than you initially expect when you start. And so the reality is that pediatric cancers have a significantly lower mutational burden. And so the data I'm showing here from Grobner et al. Is plotting the average number of coding mutations per megabase, or AKA the number of mutations in a gene across 24 different pediatric cancers and 11 adult cancers. And the take-home message from this is that pediatric cancers on average harbor around 14,000 fewer, or 14 times fewer mutations than adult cancers. And this distinction or difference is really important because fewer mutations means that there are fewer obvious drug targets using this precision oncology paradigm I just showed you. And to make things even more challenging, the pediatric cancers actually have an inherently different driver biology. So the mutations that we identify in pediatric cancers tend to be in genes that are difficult to drug directly. So while adult cancers are enriched for mutations in kinases and receptors, which traditionally have been easily targetable with small molecules, pediatric cancers are enriched for mutations in chromatin remodelers, transcription factors, and histone mutations, which have historically been considered undruggable targets. And this is where the story gets even more complicated. So recent years have seen this explosion of sequencing of patient tumors. And to give you an idea of this, I'm showing you data from a database called AACR Genie and how the samples have increased over time. And this is actually this data is out of date. I just checked before this presentation, and the current number of samples in this database have exceeded 200,000. And this doesn't even account for tumors that have been sequenced in private institutions and other databases I'm not showing you here. The problem is that we're identifying an exponentially growing number of variants of uncertain significance, or as those in the field like to call them, VUSs. And so if you look at this graph on the right in this gray bar, you can see that as we sequence more patients, this line just keeps going up and up and up, and we expect it to keep going up as we sequence more and more tumors. So without functional characterization of these variants, we cannot use these mutations to guide patient therapies. So we really just don't understand which mutations matter and which ones don't. That's really the core of the problem here. And so let me give you a concrete example in the cancer biology world. So we have this SWE/SNF chromatin remodeling complex, and this complex is very important for modifying the accessibility of DNA for various cellular processes. And it can be composed of up to 15 subunits. I'm just showing a couple here. And so if you look at this subunit as a single entity, we find that it's actually mutated in over 20% of all human cancers. And there are thousands of VUSs across all of these subunits. And for my graduate research, I focused on one of these subunits called SMARCB1, which is mutated in about 1% of all human cancers. And remarkably, if you look at the clinical characterization of the missense mutations in SMARCB1, 89% of them are classified as variants of uncertain significance, which is a problem. We don't know what any of these are doing. So that brings me to the two different projects I pursued during my graduate research. They both approach these two challenges in pediatric oncology, the variant interpretation problem and the therapeutic targeting problem, but from two different approaches. So the first part, we're trying to understand what these missense mutations in SMARCB1 are doing by systematically characterizing their functional effects using a technique called deep mutational scanning. And then in part two, we'll be tackling this therapeutic discovery problem in a pediatric cancer called Wilms tumor, which is the most common kidney cancer in children. And we'll be using a functional genomics approach called loss of function screening. All right, so we're going to start part one, and I'm going to start by giving you a little bit of background on SMARCB1. So the SWE/SNF complex binds to nucleosomes, which is the basic organizational structure of DNA, using two of its subunits, SMARCA4 and SMARCB1. Think of it like picking up a coin off of the ground. You pick it up on both sides, and that's how you hold it. And so what happens is when the complex comes in and binds to the nucleosome, it will actively shift the nucleosomes out of the way, and this increases the accessibility in these particular regions. What we see is that when you lose or completely lose SMARCB1, and this can be through multiple mechanisms, including deletions, frame shifts, translocations, or deep deletions, lots of different ways. But at its core, what happens is the SWE/SNF complex is no longer able to efficiently bind to the nucleosome. And this leads to genomic compaction across the entire genome, and this leads to lots of epigenetic and transcriptional deregulation. This ultimately leads to the initiation of several pediatric cancers with nearly 100% penetrance. Here are three that we focused on within this particular study, TRT, and RMC, all of which have pretty terrible prognoses with a 5-year survival rate of around 30%. And so the way that these tumors are typically diagnosed is through a technique called immunohistochemistry. So what'll happen is a patient will undergo a biopsy, that sample will go to a pathologist, and they'll stain that tumor sample with SMARCB1 antibody. And if the tumor comes up as deficient or there's no binding of SMARCB1, the pathologist will diagnose that cancer as SMARCB1 deficient, and that patient will be kind of categorized into that treatment category. So we know clinically that loss of SMARCB1 leads to cancer initiation, but my main question coming into the lab here was, what about missense mutations? Are these missense mutations partial loss of function, or could it be possible that missense mutations are a complete loss of function? And if that is true, these missense mutations would be missed through this diagnostic approach. And so that's kind of the motivation for this project. And so first, to study this missense variant problem, we had to establish a model system that could reliably detect pathogenic SMARCB1 variants. So we used a SMARCB1 deficient cell line called G401, which is an MRT cell line. And we introduced several SMARCB1 constructs, as you'll see through the presentation, using this lentiviral inducible system. And so my positive control here is going to be wild type SMARCB1, and my negative control is going to be a patient-derived ATRT nonsense mutation. And for those of you who aren't familiar with that, this leads to a truncated version of the protein. As you can see, there's a smaller band here, meaning smaller protein. And so what we first did is wanted to see what happens when you re-express the SMARCB1 construct in a deficient cell line. And first, we performed this qualitative proliferation assay using crystal violet over 10 days. And we see that there is a profound decrease in proliferation upon wild-type SMARCB1 expression. And this effect, or the anti-proliferative function of SMARCB1, is completely almost gone with the nonsense mutation. And so then to kind of more quantitatively approach this problem, we performed a similar proliferation assay using trypan blue exclusion over 8 days. And we saw that the wild type led to a 66% decrease in proliferation, while the nonsense mutant led to a 29% decrease. And this is a significant difference. And so this establishes that we have a reliable model system to actually detect pathogenic variants in SMARCB1. So next, we wanted to dive into the data, see if we could go find that complete loss of function missense mutation. So what we did is we went into public data sets like the ones I showed you earlier, particularly AACR-Gini and another one called COSMIC, and we wanted to find which missense mutation was being found in patient tumors most frequently. And the one we saw was this R377H mutation here on the C-terminal end. And it just so happens to be that this mutation is also found in patients with an intellectual disability called Coffin-Siris syndrome. And I will note that patients with this mutation, with this intellectual disability, do not have cancers at a higher rate than the population. So coming into this, we put this mutation through the same battery of tests where we lentivirally inducibly expressed it and performed this proliferation assay. And we saw that for the large part, this mutation actually maintained almost all of its anti-proliferative function, showing a 62% decrease, which is actually not even significant when compared to the silent, which should be acting as wild type. And so this is basically suggesting that these recurrence-based approaches are not sufficient to identify loss of function missense mutations in this particular assay. I will also note that the nonsense here, you may see as why is that also having an anti-proliferative function, and I'll come back to that later, don't worry. And so next, given that the recurrence-based approaches didn't really work, we wanted to then check the computational prediction models. And so we picked 3 leading computational algorithms, alpha missense, CAD, and REVEL. These 3 predict the functional effects of missense variants using distinct computational approaches. And it became immediately clear that all 3 models predict almost all of the missense mutations to be pathogenic. Here, I'm just showing you the percentages that exceeded the threshold that they suggested to use, or the authors suggested to use. And as you can see, alpha missense, which is probably the most popular one, it's from Google, shows that nearly 97% of missense mutations are pathogenic. That's a lot. And so CAD also predicted that 77% were pathogenic, and REVEL presented 38%. And just to really give you the extent of how many missense mutations it's showing as pathogenic, I'm showing this heat map across the coding sequence of SMARCB1 and all possible substitutions. As you can see, the whole protein's almost red. And so that was kind of confusing. We didn't expect every single missense mutation to be pathogenic. And so if you think back to the R377H mutation, I just showed experimentally that it was largely functional in this proliferation assay. Yet all 3 models predicted R377H to be pathogenic. And so this suggests that these computational prediction models are not really adequate to predict the functional effects of these mutations in a proliferation assay. And so this suggests we need to actually do experimental quantification to see what these variants are doing. And so that's ultimately what we did. So my hypothesis after looking at all this data was there must be a small proportion of missense variants that can disrupt SMARCB1 anti-proliferative function as severely as nonsense mutations, although likely through a distinct molecular mechanism. And that by so doing, it would evade detection through these standard immunohistochemistry-based approaches. And so my research goals were really twofold. I wanted to systematically characterize the functional consequences of all SMARCB1 missense mutations using this technique called deep mutational scanning. And I also wanted to identify the molecular mechanisms through which these pathogenic missense mutations disrupted SWE/SNF function and remodeling activity. So to answer this hypothesis, we used 4 distinct experimental approaches. We first used functional genomics to systematically characterize all the variants. Then we used biochemistry to interrogate SWE/SNF complex formation. Then we used biophysics to tease out the molecular mechanism driving any potential pathogenic variant. And then we used multi-omics profiling to assess various aspects of SWE/SNF biology. I'm going to walk through each of these one at a time. So for the first functional genomics approach, we use this deep mutational scanning approach, and we first had to identify 3 model systems to test these variants in. So we picked 3 different SMARCB1 deficient models spanning 3 different disease states, MRT, ATRT, RMC, the ones I brought up earlier. And what we did, previously Andy generated the SMARCB1 mutant library and introduced them into each of these 3 cell lines separately in a way that only 1 mutation was present in each cell. And this encompassed around 8,400 variants covering around 99% of all possible amino acid substitutions. And once they were integrated into the cells, they had to undergo some sort of selective pressure. So we used growth competition for this. And the idea here is pretty simple. Cells that have variants that disrupt the anti-proliferative function will increase in abundance because those cells are going to grow faster. And cells that have variants that are largely wild type are going to decrease in abundance because those cells are growing slower. It's a pretty simple concept. And so what we did is passage these cells for around 8 to 14 days, which allowed 5 doublings for each of these 3 cell lines. We harvested the cells, extracted DNA, and performed deep sequencing and functional score calculations. And so before I show you this data, it can be a little overwhelming. So I want to prime you all with 3 key questions about how I approached this particular analysis. And so, as Andy always says, the first thing you need to do before approaching any analysis is, are the controls validating? Are they doing what you need to do? And any good scientist should do that as they approach any experiment. And so I'm going to start with that. Do the controls validate? And we expect these stop codons and frame shifts to be enriched throughout this assay. And so here, what I'm showing you is the distribution of 3 different mutation types here: frame shift, nonsense, and silence. And kind of just taking a step back so you understand this, we performed the variant functional analysis using this z-score normalization method. And essentially, any mutation with a z-score greater than 2, we're considering as loss of function. And so what we saw was exactly what we wanted to see. The frame shift and nonsense mutations, which we know are pathogenic, all had z-scores centered around a z-score greater than 2, suggesting that our experiment worked, which is great. So then we plotted the frame shift and nonsense mutations over the amino acid sequence of SMARCB1. And we saw that there was this upside-down U shape, which suggested that nonsense mutations on either end of the protein are retaining partial function. And if you think back to that R377 stop data I had showed previously that actually maintained a lot of its function, this backs that up. And I'll kind of dig into in the next slide on why we think that's happening. But first, we wanted to make sure this validated in patient data as well. Are we seeing nonsense mutations in either terminus of SMARCB1 or not? So what we did is looked in another data set called CCDI, which contains a lot of pediatric cancer genomic data. And this had nonsense mutations for 13 MRT, ATRT patients. This is low, but pediatric cancer, this is pretty normal. And so what we saw was that there were no nonsense mutations observed on either terminus of SMARCB1 in the patients, which really agreed with what we were seeing in our functional data. And so kind of walking through why we think these terminus, N-terminal, and C-terminal are maintaining function. For the N-terminus, there's lots of methionines in the beginning of SMARCB1. So we think that downstream methionines past the nonsense mutation are leading to at least partial protein translation. It's picking up after the stop codon. And similarly, we actually have functional data to back this up, but we think that the C-terminal, or the right side here, maintain function because much of the protein has already been translated. So it's able to at least maintain some of its anti-proliferative function. All right, so do the controls validate? It appears yes, based on this data. So moving on to number 2. So AI is a really big buzzword right now, and everyone really hopes it's going to revolutionize how we change medicine and diagnostics and all that jazz. So we wanted to see if these prediction models were accurately agreeing with experimental data we had. And so to do this, I took the predicted functional effect for each residue across all 3 variant predictors I showed you earlier, and overlaid that on top of our experimental DMS. I'm going to refer to deep mutational scan as DMS from now on. And so what we saw was there was actually quite a significant discordance. You can see how all top 3 rows have a deep red, showing that these mutations or these residues are intolerant to mutation. This deep red is largely lost on the bottom of the experimental data, suggesting that these are not exactly agreeing with each other. And so to kind of more quantitatively approach this, we wanted to assess how all 3 mutation types are correlating with the computational predictions. And so we performed this concordance analysis between CAD, which is the only one to predict for all 3 types of variants. It's a little bit of a detail, but we can only do this with one. And we compared it to our experimental data. And we saw that for the most part, silence and nonsenses were concordant. Although I will point out this cluster here are nonsense mutations in the terminal regions. So CAD was unable to pick up on this kind of residual function for these terminal nonsense mutations. And where the discordance was most pronounced was with these missense mutations. As you can see here, many missense mutations in gray showed they were pathogenic within CAD. However, within our experimental data, only a small proportion were actually shown to be loss of function. So altogether, this data shows that, yeah, the computational prediction models don't really predict the effects of missense variants in this assay. So to answer number 2, no, they don't really. And so next question, number 3, is SMARCB1 generally tolerant to missense mutations? You probably gathered it from the previous slide, but the answer is yes. It appears that SMARCB1 is generally tolerant to the vast majority of missense mutations. And so the heat map that I'm showing here is showing the functional scores for every possible residue and substitution within SMARCB1. And what we can see here is that the vast majority of these missense mutations are being tolerated, shown through the light blue and light red shading. Only a small proportion actually were shown to be loss of function, and these are highlighted in this kind of black box up here. And so this was initially surprising. SMARCB1 is a tumor suppressor with a pretty well-defined role as a driver of cancer. However, it's largely tolerant to missense mutations. And so we went looking through the literature, and this likely reflects the fact that SMARCB1 is a scaffolding protein. As long as the overall structure of SMARCB1 is maintained, it's able to tolerate the effects of missense mutations. And this effect has actually been observed in other kind of scaffolding proteins within the ribosome as well. And so when I applied this threshold of a z-score greater than 2 to the missense mutations, we saw that only 1.4% of missense mutations actually reached loss of function within this assay. And then when we plotted them across the amino acid sequence of SMARCB1, we saw that there are specific regions that were enriched for these loss of function missense mutations. And this begged the question, what are the structural regions that are enriched for loss of function mutations? So just to come back, yes, to answer the last question, it's generally tolerant to mutations. And so what we did here is we wanted to see which residues were most intolerant to mutations across the board. So we averaged all possible 19 amino acid substitutions for each residue and then just ranked them. And so what we identified was that there were 13 intolerant residues that reached significance. 6 were in the winged helix domain, 3 are in the IDR, and 4 are in the repeat 2 domain. I'm going to focus on these 4 in the repeat 2 domain for the rest of the talk. I will also note that the most recurrent residue here actually didn't even reach significance, which is, I think, quite interesting. And so When we mapped these scores on top of the structure of SMART-B1, we saw that all four residues that came up as top hits were actually closely interacting within three-dimensional space. This suggested that they may be forming a hydrophobic network within repeat 2, and that could be being disrupted. And I'll come back to that in much greater detail later in the slides. And so, yes, they're interacting. And so next, okay, great, we did the screen. Now we need to validate if these top hits actually are validating in that same proliferation assay I showed you earlier. And so check, we finished the first approach here. Now moving on to biochemistry. So first, before we move on to that though, we needed to validate these. And so as I showed you before, we're using this lentiviral inducible system, and I designed the top hits from each of these four residues with a positive silent control and a negative nonsense control for each. And what we saw was there was robust SMARCB1 expression for each of the missense mutations. And then we performed the same qualitative proliferation assay and saw that all four mutations had a noticeable decrease in anti-proliferative activity, as you can see here, 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 and here. And then we performed the same quantitative assay for each of the different conditions shown here. And just to kind of contextualize all this, I'm showing you the mean decrease from the previous experiments I have talked about in this presentation. And I think take-home message is that all four missense mutations led to an increased, they eliminated anti-proliferative function greater than the R37 recurrent cancer mutation. I think most notably, the missense mutations 281P and I315R decreased anti-proliferative function equal to or greater than the patient-derived nonsense mutation, despite having SMARCB1 protein present. And so given that we've established this validates in an assay, we wanted to see what is the structural basis for this loss of function. So to do this, we wanted to ask the question, what is happening to the SWI/SNF complex in the presence of each of these mutations? So first we had to isolate nuclear protein and biological triplicates, which was super fun. And then we then we performed SMARCA4 IP mass spec. And so the reason we're using SMARCA4 for the bait here is because SMARCA4 is the core ATPase subunit of SWI/SNF, which is present in all the SWI/SNF complexes. We'll kind of come to that in a second. And so what we saw when we performed quantitative IP mass spec was that when comparing the mutant to the wild type, or mutant to the silencer essentially wild type here, we saw that there was significant depletion of a lot of SWI/SNF subunits. This suggests that these mutations are destabilizing the SWI/SNF complex. And if you look at these subunits that are similarly lost in both mutations, you see that all three replicates showed depletion of these subunits that were shared across each mutation. Notably, the important ones that are core structural features of SWI/SNF are ARID1A, SMARCC1, and SMARCE1. These are really important subunits that are being lost. And so my next question was, are there SWI/SNF subfamily specific losses? And so a lot of you are probably like, what are you even saying here? So to give you some background, the SWI/SNF complex exists in three distinct subfamilies, and they all have distinct genomic roles. So the first one, called CBAF, or canonical BAF, is known to bind to enhancers and performance remodeling activity there exclusively almost. And then you have PBAF, which is predominantly responsible for remodeling at promoters. And then you have the probably least studied GBAF or NCBAF, which is responsible for remodeling at CTCF sites and promoters. And so to see how each of these subfamilies are being affected, we overlaid the significantly depleted data from the mass spec on top of these kind of three subfamily categories. So what I'm showing here are all the subunits included in each subfamily. The ones that are specific to that particular subfamily are hashed. The ones without hash are kind of common amongst all of them. The ones that were significantly depleted are shown in orange. And what I want you to take home from this graphic here is that CBAF was being predominantly affected. As you can see, all three specific subunits were being lost. PBAF, we saw one of four subunits being lost, but it seems that three were at least maintained. And GBAF, which actually doesn't even have SMARCB1, we didn't see any subfamily specific loss. So this suggests that these mutations are primarily affecting CBAF stability, and this suggests that it's likely distal enhancer regions that are being affected. And so next, we wanted to kind of tease out the molecular or atomic basis for this complex destabilization. And so check, We finished that one. Now we're going to move on to the biophysics part. A lot of you are probably like, what even is biophysics? Okay, so that was me when I first started this too. So why these mutations are causing this destabilization. We collaborated with Dr. J.C. Gumbart's lab at Georgia Tech to really hone in on how to run these molecular dynamic simulations. And here, I hope this works. We have a video that shows what molecular dynamic trajectories actually look like. You're essentially watching how the protein moves over time in a solution. And so there are several benefits to molecular dynamics compared to just typical cryo-EM studies. First, it reveals how proteins move and flex, not just these static snapshots we get from cryo-EM. Second, it shows how a mutation can change protein flexibility and its stability at a quantitative level. And lastly, it can uncover hidden mechanisms that are hidden within static structures. And this is really important because AlphaFold just predicted the structure of almost every protein in the human genome. And so you can apply this to really any protein you want. So it really recently became a very powerful technique. And so before I dig into the analysis, I kind of need to walk through how the repeat two domain of SMARCB1 actually is set up. So you have two beta sheets shown in pink here that are opposite of two alpha helices. And these are binding to each other through stabilizing hydrophobic interactions. And so we wanted to see how these mutations were disrupting this hydrophobic barrel core. And so across 12 replicates, we calculated the amount of energy that was being introduced into the system with each of these two missense mutations. And we saw that on average, there was around a 5 kilocal per mole increase in energy within these two colored regions. For those of you who are not familiar with biophysics, this is a very substantial amount of energy increase within this system. And so then trying to understand which residues may be driving this increase in energy, we performed this contact frequency analysis. And we saw that there were lots of different residues that were losing interactions under 4 angstroms, but that's not really giving us a quantitative approach of what's going on here. So the next question was, what are the specific residues driving these unfavorable interactions? So what we did next was these residue level energy calculations. And really the take home message, what we saw here is that residue W281 sits at the core of this important stabilizing network within repeat 2. We saw that 281 was having interactions with other subunits within the repeat 2 domain, notably 290, 315, 319, and 293. And we saw that with both W281P mutations and I315R mutations, they disrupted these stabilizing interactions to an extent of almost 2 kilocalories per mole, which again is a huge increase in energy. I will note these are probably acting through different mechanisms. The proline being introduced here likely is unable to perform these hydrophilic bonds with the other residues. And this I315R is introducing a positive charge into a hydrophobic interaction, which is likely causing kind of a repulsive force due to the charge. So now we have, I think, what is a decent mechanism for why SMARCB1 is leading to the destabilization of SWSNF. It's likely propagating throughout the entire complex through this repeat 2 domain destabilization. We next wanted to ask, how do these structural perturbations disrupt SWSNF remodeling activity? And so that brings us to our last experimental approach here, which is multi-omics profiling. And so for those of you who aren't super familiar with modern sequencing technologies, I'm going to explain them with an analogy. So imagine your genome is like Atlanta. You have in Atlanta roads that are typically backed up with traffic all the time. And you also have roads that are constantly under construction, like Spaghetti Junction or something like that. Similarly, your cells have regions that are constantly accessible and constantly closed, kind of like always maintaining this closed and open kind of shape. And similarly, there are regions of the genome that are constantly under construction, aka having SWSNF or other remodeling factors bound to them. And so imagine I'm a traffic reporter trying to figure out which roads have traffic and which roads have construction sites. Typically, I would just ask Google Maps, but unfortunately, cells are not that cooperative, or I would have saved like 2 years of my PhD. So scientists have developed 2 techniques to answer this question. First, a taxi kind of acts as your traffic map. It'll show you which roads are jammed with traffic and which ones are open. Or in other words, which regions of the genome are accessible and which ones are closed. And the second technology, called cut and run, will tell you where these construction sites are actually working. Or in other words, which regions of the genome are being bound by these remodeling factors. I'm simplifying a lot here, but I just wanted you guys to be with me as I present the rest of the data. And so when I looked at the traffic map of the cell or the accessibility data based on ATAC-seq data, we saw that there was a pretty remarkable phenotype in the presence of each of these mutations. We saw that there was almost a third of typically accessible regions that had lost accessibility, particularly these sites right here. And this is the equivalent of these mutations causing traffic jams in around a third of the city of Atlanta. It's a pretty profound difference. And so when we came into this experiment, we kind of knew that the nonsense mutation was going to be a 5-car pileup on the connector. It was going to cause a lot of traffic. We didn't really know what to expect with the missense mutation. And so it could have just been like a fender bender or maybe a slight wreck, but no, the missense mutation was phenocopying the nonsense mutation with the same 5-car pileup. And that was a really shocking finding. And so we next wanted to ask the question, which genomic regions are actually being disrupted? Or if you want to keep the analogy going, which roads are having traffic? And so what we saw when we annotated which genomic regions were being affected was that it was largely distal, likely enhancer regions. As you can see through on the right bar here, a large gray and large black here. And so when we kind of dug more into these genomic annotations and tried to find which transcription factors were binding or were most enriched in these affected sites, we saw that the affected sites were highly enriched for this AP1 transcription factor binding motif. Among the sites that were unaffected, we saw that CTCF sites were largely enriched. So this data suggests that SMARCB1 missense mutations disrupt AP1 binding sites or the accessibility at these sites, but largely spares accessibility defects at these CTCF sites. And so then, having this answered, we wanted to ask the next question, are these remodeling defects being driven by loss of SWE/SNF occupancy or loss of SWE/SNF activity? And if we want to come back to the metaphor, this is kind of like, are the construction crews still there? They're just on lunch break? Or are the construction crews packing up and going home? And so that's kind of like the two different questions we wanted to ask. So to do this, we perform cut and run, and we perform this for 3 different subunits within the SWE/SNF complex, SMARCA4, SMARCB1, and SMARCE1. And what I'm showing here is it's a subset of the accessibility data I just showed. I'm only showing the unaffected and affected sites that were also bound by SWE/SNF. So it's kind of like a smaller number, but generally the same regions. And so I'm going to walk through each of these one subunit at a time. And so what we saw when we looked at SMARCA4 occupancy was that while across every region there was a slight decrease in SMARCA4 occupancy, we saw the most predominant loss at this affected region, suggesting that the SWE/SNF complex was no longer binding, aka the construction crews were going home. They weren't even there anymore. And so then we wanted to look at how SMARCB1 was binding, and this was actually quite interesting. So it seemed that both mutations had a mutation-specific effect. And if you remember, this is the mutation, this is the protein the mutation's actually in. And so we see that with the W281P mutation, this leads to almost a global loss of SMARCB1 binding at both the unaffected and the affected regions. Conversely, with the I315R mutation, we see that there's almost this selective loss. At the unaffected regions, we see that binding is largely maintained. However, at these affected regions, we see that binding is almost completely lost. And so that was just interesting to see those mutation-specific effects, but ultimately the remodeling activity was pretty much the same for both. And so then last, this brings us to SMARCE1, which was, I think, probably the most interesting finding from this study. We saw that there was greater SMARCE1 occupancy loss in the missense mutations than in the nonsense mutations. And so it's not entirely clear how these unaffected sites are maintaining accessibility despite the fact that SWE/SNF isn't even really bound there anymore. If I had to hypothesize, it's probably because of redundant remodelers such as CHD or ISY. But nonetheless, we did see reduced occupancy at the affected sites as well. So altogether, these data suggest that this is loss of SWE/SNF occupancy, not loss of SWE/SNF activity. And so then lastly, this is the last thing for this project, is we're trying to see if all of these defects I've been showing you are actually leading to downstream transcriptional effects. And that's really what matters in the cell. So what we did here is performed RNA and attack concordance analysis. And I'm simplifying a lot here, but I essentially took the sites that had significant changes in accessibility and mapped those sites to the nearest gene. And we just wanted to see if increased accessibility led to increased transcription and kind of vice versa. And that is really ultimately what we saw. We saw that regions that had an increase in accessibility had an increase in transcription. And this effect was largely lost within the missense mutations. So these defects are leading to transcriptional defects as well. All right, so we finished all 4 approaches, and this means we can kind of wrap up this part with a mechanistic model. So in wild-type cells, or wild-type SMARCB1 cells, you have like a normal repeat 2 domain. It's stable, and it's able to integrate efficiently into the CBAF or PBAF complex. These are able to efficiently bind to these AP1 distal sites and perform their normal remodeling function, and this leads to normal antiproliferative function. Conversely, in both mutant contexts, it leads to the destabilization of this repeat 2 domain. Ultimately, this leads to destabilization of the entire Sweet-Sniff complex, although the exact mechanism is undetermined. Then, because of this destabilization, the Sweet-Sniff complex can no longer bind to these distal AP1 sites and can't perform its normal remodeling function, and this leads to loss of antiproliferative function. So in conclusion for this part, this is the first study to show that missense mutations are functionally equivalent to nonsense mutations in SMARCB1. Kind of walking through kind of more sub-conclusions, I think one of the most interesting thing is that computational prediction models are not always accurate for context-specific functional readouts. So for instance, if we focus on the R377H mutation, it's clearly pathogenic within an intellectual disability context. However, it's not pathogenic within this proliferation context. So I think we kind of need to rethink how we assign the term pathogenic to missense variants. I don't think it's this binary on and off switch. I think it's very context dependent if it's pathogenic or not. So that's something to think about. Second, we see that missense mutations can lead to the destabilization of the entire Sweet-Sniff complex. This has never been reported before, and this leads to global chromatin occupancy and remodeling defects. Similarly, we show that these intramolecular forces appear really important for structural scaffolding proteins, such as SMARCB1. I'm excited to see if this applies to other structural scaffolding proteins as well. I think probably the most clinically relevant finding from the study is that it challenges the diagnostic reliance on immunohistochemistry to diagnose these cancers. If you have a non-functional SMARCB1 protein in these tumors, they're going to come up as SMARCB1 retained, but this patient would have a very aggressive course of disease. And so I think this suggests we should be doing more targeted sequencing on patient tumors. All right, so I swear this part's going to be a lot shorter, but we're going to shift gears to a different type of cancer called Wilms tumor. And so this is the most common pediatric kidney cancer, which accounts for around 6% of all childhood cancers. And it typically occurs in children who are around 3 years old. And due to optimization of chemotherapeutic regimens, we see an impressive 5-year survival rate of 90%. Despite this 5-year survival rate being so high, there are definitely still challenges though. And so this cancer can be categorized into 2 histologies. You have favorable histology and diffuse anaplastic. Diffuse anaplastic has a much worse prognosis with around a 5-year survival rate of 50%. And based on stage at which a patient presents, they're categorized into 2 different treatment categories. You have, for low-risk patients, surgery plus chemotherapy with vincristine and dactinomycin, and high-risk patients, in addition to those, also get doxorubicin and radiation. So despite all these advances, 15% of patients have recurrence. And I think most importantly, there are no effective targeted therapies. Wilms tumor relies entirely on chemotherapy, surgery, and radiation for its therapeutic approaches. So there is an urgent critical need to develop targeted therapies in this cancer. And that is really what motivated this study. And kind of historically, the reason why we don't have targeted therapeutic approaches is because a lot of the cell lines that were used in the past ended up actually not even being Wilms tumor. The ones that we thought were Wilms tumor ended up being rhabdoid tumor and Ewing sarcoma, so those studies were pretty much not applicable to this cancer type. More recent attempts have tried designing new Wilms tumor cell lines, but finite passaging has limited the utility for these genome-wide loss of function screens. And there are organoids and PDX models, but these are not sufficient for these therapeutic drug testing studies. And so our approach here was to develop new faithful Wilms tumor cell lines that actually could be passaged. We wanted to validate that these faithfully recapitulated Wilms tumor both genomically and transcriptomically, and then perform loss of function genomic screening. And so kind of just walking through what we first did, we developed these 12 new patient cell lines from Wilms tumor patients, and we performed whole exome sequencing. And we wanted to make sure these had mutations you would expect in Wilms tumor. And so kind of just walking through here, loss of heterozygosity at chromosome 1p and chromosome 16q are classic Wilms tumor markers, and we see that in some of our samples. Similarly, we saw that there were mutations in W2T1, beta-catenin, and drosha. And we also saw that there was copy number alterations, or notably 1q gain. All of these are very classic Wilms tumor mutational signatures. And so this confirms that at least genomically, these represent Wilms tumor. We also wanted to see if transcriptomically they also represent Wilms tumor. So what we did is performed bulk RNA sequencing and performed UMAP clustering on top of other public data sets that have childhood patient tumors, spanning, 4 different cancers here. And what we saw when comparing to these public databases, Target and St. Jude in particular, is that our Wilms tumor cell lines in red here overlaps very cleanly with these publicly published Wilms tumor cell line, or Wilms tumors. These are not cell lines. And we also saw that there was upregulation of Wilms tumor specific markers, notably 6.2 and cited1 as compared to the normal cell lines. And so now that we have these faithfully described Wilms tumor cell lines that actually match patient tumors, we wanted to perform these functional loss of function screens to identify synthetic vulnerabilities in Wilms tumor. And just kind of briefly going over this, it's a very similar idea to the previous screen, but we performed this orthogonal RNA and CRISPR loss of function screens in a way that each cell uptook one single guide RNA or one shRNA. And this encompassed around 400, not around, it is 429 genes. And the idea is that cells that took a guide RNA that was bad for its proliferation, that would decrease in abundance, and ones that didn't affect proliferation would increase or stay the same. And so in this case, we're focusing on ones that are depleted, not enriched, as compared to the last study. And we did this in 5 unique Wilms tumor cell lines, and we thought that high confidence hits validated by both methods would be good therapeutic approaches. And so when we looked at the data from both methods in all 5 cell lines, we saw that XPO1 came out as a top hit amongst 7 other genes. However, XPO1 has targeted inhibitors that are already being used in the clinic, particularly for multiple myeloma. So this seemed like an immediately actionable target. So first, we wanted to see how these tumor cell lines were responding to an XPO1 inhibitor called KPT-330. I'm just going to call it KPT because it's a mouthful. So when we treated the tumor cell lines with KPT and the normal cell lines with KPT, we saw that there was a 6-fold increase in sensitivity in the tumor cell lines as opposed to the normal, which suggests that this is a tumor-specific vulnerability. We also wanted to check if we had on-target activity. So we first looked at XPO1 levels and saw that they were reduced upon KPT, which is a reported mechanism for this drug. Similarly, we also saw an increase in abundance of P53, which is an important tumor suppressor, and this is also a known mechanism of KPT. Interestingly, we saw downregulation of this mitotic spindle factor called TRP13, and we'll come back to that in just a second. And so next we wanted to see how KPT affects the cell cycle within these Wilms tumor cell line models. And we saw through 2 different methods that it increased G2M arrest. And so on the left here, I'm showing flow cytometry data showing an increase in G2M as seen through this blue bar, and this was significant. And transcriptomically, we saw that we had downregulation of many G2M checkpoint genes. And importantly, this was actually a P53 dependent sensitivity. When we knocked out TP53 using CRISPR, we saw a 20-fold to 15-fold increase in resistance to this drug. And so trying to tease out a little bit of mechanism here of how KPT is mediating these effects, we looked into the transcriptional downregulated targets, and TRP13 stood out to us. So we further studied that TRP13 gene by performing TRP13 suppression in these Wilms tumor models. And we ultimately saw that it recapitulated a lot of the effects that we saw with KPT treatment. So for instance, when we knocked down TRP13, it led to around a 50 to 80% decrease in cell viability. We also saw that TRP13 knockdown led to downregulation of G2M checkpoint genes, notably cyclin D1, which is a very important promoter of cell proliferation. And lastly, when you look at the overlap of downregulated genes between KPT treatment and TRP13 knockdown, we had 66 overlapping genes, and among those, they were enriched for regulation of cell cycle. And so this suggests that TRP13 at least partially mediates the effects of XPO1 inhibition. And this is the last slide, I swear. And so we next wanted to see if this KPT drug would be synergistic with any other standard of care chemotherapeutics being already used in the clinic. So we tested it with actinomycin, vincristine, and doxorubicin, and we saw that there was synergy only present with co-treatment with doxorubicin and KPT. And so with this as kind of our target, we then applied this combination drug treatment across all 12 cell lines that we had developed. And we saw that all 12 Wilms tumor cell lines had either additive or synergistic effects when treated with both of these drugs. Importantly, this synergy was not found in the normals. It was actually antagonistic, again, suggesting this is a tumor-specific selectivity. And I think most importantly here, when we bring this to an in vivo setting, we saw that co-treatment with KPT and doxorubicin led to increased survival for mice with Wilms tumors. And so in conclusion, this is the first study to use loss of function screens to identify targetable dependencies in Wilms tumor. We established 12 Wilms tumor cell lines. This is not a trivial task. We also provided evidence that nuclear export inhibition is a tumor-specific vulnerability for Wilms tumor and showed both in vitro and in vivo synergy. And I think something that's very exciting for me is that there's actually an ongoing phase 2 clinical trial testing nuclear export inhibition in recurrent and refractory Wilms tumor, and this was supporting evidence for initiating that trial. So with that, I will end. I want to say thank you to a ton of people. I'll get more into this later, but walking through here at my lab, you guys are absolutely incredible. Couldn't have done this without any of you guys cheering me on the entire time. Obviously, Andy, thank you. You're the best, but I'll get to you later. My committee, thank you for hearing me over the years and giving me advice and just being generally pretty awesome. I want to thank Preetha. I don't know if she's on the Zoom, but she was really critical in helping me with this mass spec IP experiments, and I really cannot thank her enough. I want to say Sakina Hicks, you helped with the ATAC-seq data. I don't know, you're probably not on here, but thank you to her. If you need ATAC-seq help, she's awesome. And thank you to my funding, both the T32 for supporting me for the first 2 years and the NCI F31, which supported me up until now. So if I'm forgetting anyone, I'm sorry, but we'll get to you. Thank you.